Good day. I am Andrea Añonuevo, and today I'll be presenting my thesis entitled In Silico de Novo Drug Design of a Therapeutic Peptide Inhibitor Against UBE2C in Breast Cancer. I will be presenting for this paper my introduction, objectives, methodology, results and discussion, as well as conclusions and recommendations. The World Health Organization declares breast cancer as the most prevalent cancer in the world, with 7.8 million women being diagnosed with the disease in the past 5 years. For breast cancer, inherited genetic mutations in specific genes as well as a family history of breast cancer increases the probability of breast cancer development in women. However, WHO posits that approximately half of breast cancer cases develop in women who have no identifiable risk factors aside from being female and being over 40 years of age. Currently, the most common method for cancer therapy is targeted therapy. The key component in these targeted therapies is the presence of a definitive biomarker specific to the site of cancer within the tissue, such as in the case of luminal breast cancer and HER2-positive breast cancer, which exhibits high levels of certain proteins. However, there is a third cancer subtype which is characterized by its lack of defining biomarkers, which is why it's called triple-negative breast cancer. This makes it hard to approach using targeted therapy methods. This is why the standard of care treatment or SOC treatment of triple-negative breast cancer is still conventional chemotherapy treatments. There is a definite lack of options when it comes to treatments for TNBC, specifically minimally harmful treatment options, unlike with the other two breast cancer subtypes. Recently, protein-protein interactions or PPIs have been studied as possible targets for drug action. PPIs are key components of cell survival and thus disease progression through regulation of signaling pathways such as replication, transcription, and translation, which is why PPIs have become very attractive prospects for drug action. In relation to this, UBE2C is one of the most widely studied E2 targets for cancer therapy. It was found that UBE2C is overexpressed in breast cancer patients and is associated with risk of death in breast cancer. Studies show that inhibiting UBE2C expression in BC cancer cell lines reduced cancer proliferation rates and metastatic or cell invasion abilities of the cells. However, PPI interfaces are significantly larger than binding areas that small molecule drugs are able to cover. Thus, it was found that peptides are more naturally suited to the large or flat surfaces found in PPI interfaces. Additionally, they are great drug candidates due to their low molecular weight, high flexibility, and minimal toxicity. In addition, peptides have also shown to have better pharmacokinetic properties as compared to small molecule drugs. This study aims to design a peptide inhibitor to disrupt a UBE2C regulated pathway in breast cancer in silico, specifically to generate a short anti-cancer peptide inhibitor, to evaluate its performance as a drug via docking studies and molecular dynamics simulations, as well as to test the safety of the drug using immunogenicity and amyloidogenicity tests. This study is conducted to verify whether peptide drug inhibitors are effective treatments against breast cancer via inhibition of the ubiquitin protein UBE2C. It is also conducted to expand the catalog of breast cancer drugs, as well as to provide an additional in silico framework for the possibility of using peptide drugs against cancer by targeting the protein UBE2C. This study was conducted by first, data acquisition via protein interaction prediction using protein interaction databases, specifically using String, IID, UniHi, BioGrid, and UHURI. Peptide design was then done by hotspot residue prediction, then subsequent sequence similarity searches to generate hit sequences, after which the hit sequences are further modified in order to increase its affinity with UBE2C. After gathering the lead peptides, the PepFold3 server was then used in order to generate the complete peptide using the lead sequence input. In addition, the PepFold3 server is also able to simultaneously run docking simulations with the predicted peptide structures. The resulting complexes were then evaluated using the pizza-protein interaction Z-score assessment system. The docked peptide then underwent evaluation using molecular dynamics simulations and MMPBSA calculations using the GROMACS suite, where the MD simulation was run for 10 nanoseconds. In addition, biopharmaceuticals must undergo immunogenicity and amyloidogenicity studies to determine the possibility of the protein to elicit adverse immune responses from the patient's body. This will be done using the following web servers. Before anything, a PPI target must first be established. The PPI target must be functionally relevant to the disease pathogenesis pathway of breast cancer and must have extensive research and evidence available. Consensus shows among all the databases used was that two proteins in the anaphase promoting complex subunit family were the top hits for interacting partners with UBE2C. In the end, ANA-PC2 or anaphase promoting complex subunit 2 was chosen over ANA-PC11 due to its functional relevance in the cell cycle pathway, particularly the mitotic pathway, as well as the immediate availability of its protein complex structure with UBE2C in RCSP-PDB. Shown here is the NMR crystallization structure of the ANA-PC2 UBE2C protein complex. The peptide design was done by determining binding residue hotspots in the PPI target complex using the butylalanine scanning web server, which was used to conduct interface alanine scanning and cluster residue hotspot prediction, which was used in order to determine which cluster of residues cooperatively contribute the most to the interface binding energy. The shown interface contains the residues L753, Y757, M761, N764, L765, M776, and F780, which were located within ANA-PC2 and cut out to form the clamp-plate strand. In addition, a literature search was done in order to determine the binding site of ANA-PC2 to UBE2C. Using the fragment as the query sequence, several template sequences were made by doing sequence similarity searches across several peptide databases, from which only the Conno server database yielded hits. In the connotoxin peptide server, the top hit was a conantokin T20 peptide, and CBI BLAST was also used in order to conduct a similarity search against the entire PDB database. Specifically for this query, proteins from human species were excluded, and hit sequences with the lowest number of gaps were taken. The BLAST query yielded two more lead sequences from the proteins putative S-adenosyl methionine-dependent methyltransferase from Listeria monocytogenes and DNAD-like replication protein from Streptococcus mutans. To ensure its binding affinity with UBE2C, the sequences were modified slightly by replacing certain amino acids with hotspot amino acids in their correct sequence position. After preparing the lead sequences, PepFold3 web server was used to conduct both the peptide structural prediction and the protein peptide docking. All three lead sequences were run through the PepFold3 web server, which runs through 100 separate simulations and generates a model with the different binding posts for each run. For the purposes of the study, only the top 3 dock models for each lead sequence was taken. The dock models were assessed using a protein interaction assessment server PITSA. The results of the PITSA assessment were compared with the results for the reference PPI complex, which also was run through the PITSA server. From there, only the models which surpassed the score of the control complex were considered. In addition, due to the flexible nature of the docking server, only models wherein the peptide was bound within the correct binding pocket were considered. For the binding post evaluation, the docking conformations resulting from PepFold3 are then structurally aligned with the UBE2C and a PC2 complex, which shows the binding pockets identified using the F-pocket server. The results are then analyzed to determine the validity of the binding post for each dock model. Through this assessment, only one was chosen to be run through molecular dynamic simulations due to computational load restraints. Thus, the second binding conformation for P20M was chosen as it was the model with the highest protein interaction score. Shown here is the doc-PDB structure of T20M. A molecular dynamics simulation was done in order to validate the binding conformation of the chosen candidate peptide in an aqueous solution. From the figure shown above, the root mean square deviation, or RMSD, of the T20M UBE2C complex remains stable at a value comparable to the reference RMSD. This indicates that the structure remains stable throughout the entire 10 nanosecond run. The radius of gyration measures the compactness of the given protein structure, where it is known that alpha helixes result in a lower radius of gyration than beta sheets or structures with a combination of both. This explains why the T20M complex has a lower radius of gyration, as a T20M peptide is composed of a single alpha helix. The potential energy plot during energy minimization simply shows that energy minimization steps worked correctly and was able to relax the input complexes. In the following graph, the potential energy during the MD production run, as illustrated per picosecond, simply denotes whether or not the structure was energetically stable throughout the entire run. Using the H-bond command in GROMACS, the amount of hydrogen bonds for every second of the simulation may also be observed. Comparing the two graphs, it can be seen that the T20M complex exhibited less hydrogen bonds than the reference complex during the simulation. MMP-BSA calculations were carried out using the G_MMP-BSA tool in GROMACS. This was used to determine the binding free energy of the candidate complex and the reference complex. It can immediately be seen that the binding free energy values of the two complexes are different. Generally, the more negative the value is, the better, as this indicates better stability and a tighter bound conformation of the ligand to the protein active site. Thus, it can be seen that there is a large disparity between T20M and NAPC as interacting partners with UBE2C. There is also a large electrostatic energy difference between T20M and NAPC2. This may be due to the obvious difference in amount of hydrogen bonds formed by T20M and UBE2C as seen previously. Upon visualizing the complex in Chimera X, the interface revealed a distinct lack of hydrogen bonds that may strengthen the binding of T20M to UBE2C. On the other hand, upon inspecting the interacting residues of NAPC2 to UBE2C, it can be seen that T20M complex lacks the crucial hydrogen bond interaction between asparagine 764 and serine 51 and threonine 65 at the binding interface. This is important because hydrogen bonds between proteins and any ligand provide direction and specificity into the interaction, which increases the binding affinity between the two molecules and provides selectivity in the interaction, which is crucial in creating ligand therapeutics. Finally, peptide immunogenicity and amyloidogenicity predictions were determined to see if the proposed peptide inhibitor candidate has a tendency to form amyloid fibrils and induce an inflammatory immune response. The PASTA 2.0 server has its own scoring algorithm which measures whether or not a protein will form amyloid fibrils. The prediction was carried out with a 90% confidence setting in the server, which has an aggregation energy threshold of negative 2.8 PEU. Values that are more negative than the threshold indicate the probability of protein aggregation. Thus, from the results shown above, the UBE2C T20 complex is predicted to not exhibit any amyloid fibril formation. Overall, the results indicate that while T20M is not prone to protein aggregation, T20M may be classified as immunogenic, as in able to elicit an immune response from the body. This is a major problem in protein-based therapeutics, and while peptides are not typically immunogenic, it is still a concern when talking about therapeutic molecules. Thus, it can be concluded that the protein NAPC2 was chosen as the PPI partner due to its known role in facilitating cell proliferation and mitotic progression. Initial docking results show promise of a viable peptide drug and has a stable binding affinity to UBE2C. However, binding free energy calculations suggest further modifications are needed to the peptide to increase its binding affinity. The author recommends further analysis of additional peptide drug candidates, increasing the MD production run length to 100 nanoseconds to get better resolution results, as well as MMP-BSA calculations. Further modifications to the lead peptide may also enhance peptide binding affinity to UBE2C. That is all for my presentation. Thank you and stay safe.